Hello everyone, welcome back to this educational channel called Learn Bio with Janet. Today we'll be doing a chapter assessment for chapter 6, and I call this the Mitosis challenge. So you can test yourself by trying the questions in this topic. But first of all, let's look at what we should have studied until now. These are all the videos that have been produced so far on chapter 6, cell division, for the KSSM Form 4 syllabus. So in the first video, 6.1, we discussed the terms used in cell division, such as somatic cell, gamete, diploid, and haploid cells, and so forth. In the second video, 6.2, section 6.2, we studied the cell cycle. After that, it was followed up by 6.2, mitosis part 1, where we discussed why mitosis is needed, the function of mitosis, the importance of mitosis, and where it occurs, and also the application of mitosis in agriculture and other fields. In 6.2, we discussed the process of mitosis. So this is an important video for you to go through, where we studied the 4 stages of mitosis: prophase, metaphase, anaphase, and telophase. After that, there was an animation on the process of mitosis to reinforce what we have studied about the process of mitosis. So this short animation is very helpful for you to go through as an overview of what we studied in mitosis part 2. Next, we had this topic of cytokinesis, which is the process that follows mitosis, and it's the division of the cytoplasm. So in this video, you're going to assess yourself to find out, evaluate yourself, to find out whether you've understood what you've studied so far in the previous videos. So my advice to you is that please make sure that you've gone through all the videos before you come to the assessment, and then only then you are prepared to try the assessment. If you do not want to take the assessment, then you can just go through the questions and answers as they are here, right? And in the next video, we will talk about another type of cell division called meiosis, and that will be subtopic 6.3. All right, so this is for the next video. Before discussing the exercise, let's revise through the various phases of mitosis. Now, before mitosis, there's interphase. In interphase, the chromosomes exist as long, thin, thread-like structures called chromatin, which are not clearly visible, under the microscope. Then, as the cell goes into prophase of mitosis, the first stage of mitosis, a few things occur. So think of CNS squared. Remember CNS squared, or pros and cons squared. C is for chromatin, shortened and thickened to form chromosomes. The other C is for centrioles. Centrioles move to opposite poles. Now the N is nuclear membrane, correct? Nuclear membrane disintegrates. Another N is nucleoli or nucleolus. Nucleolus disappears. The S is for what? Spindle fibers. Spindle fibers start to form. And the last S is not an event, it's just an observation. Each chromosome is made up of 2 sister chromatids joined at the centromere. Okay. Now next, in metaphase, chromosomes become aligned at the equatorial plane. That's the main event. Align means line up in a straight line. Okay. Now then, in anaphase, the spindle, firstly the centromere will divide. Secondly, the spindle fibers will contract. And as they contract, they will pull the sister chromatids apart. One sister chromatid to one pole, the other sister chromatid to the other pole. So sister chromatids will separate and move to opposite poles. Okay. And lastly, in telophase, chromatids arrive at the opposite poles. The nuclear membrane is formed again. Nucleolus is formed again. And the chromosomes, they will uncoil and become the fine thread-like chromatin once again. Okay, that's all for the different phases of mitosis. That is prophase, metaphase, anaphase, and telophase. Okay, I hope you have drawn the diagrams which we have discussed in the second video. Here is the first question. So you can start to evaluate yourself, and you can take out a pen and paper and try to find the answers, so that after a few minutes you can come back to the same question and we'll discuss the answers. You can pause the video, you can screenshot the diagram, pause the video, you can go through the questions and then write your answers on a piece of paper. Alright, after that I will discuss the answers and you can mark your own answers. In that way you can evaluate yourself, you can evaluate your memory of this topic and also your understanding of this topic. Another way to do it is just to follow along and just listen to the comments and the answers given. Alright? Okay, so I will give you, say, a few minutes to do this question, the first question, and after that we'll discuss the answers. So this question should not take more than 3 to 5 minutes. Okay, now the question is like this: identify the phases A, B, C, and D of mitosis and arrange them in the correct sequence. Okay, so you can screenshot the question and pause the video for a while. Here I will wait for about 10 seconds, then I will give the answers. Okay, let's go through the answers. Identify phases A, B, C, D. Phase A is when the chromosomes the chromatins start to shorten and thicken to form the chromosome structures. So this is prophase, right? And then B, you can see 2 cells beginning to form and 2 new nuclei already forming. So this is the end stage of mitosis. The last stage is telophase. C, we can see the sister chromatids separate and move to opposite poles. This is anaphase. And D, we can see the chromosomes aligned or arranged at the equatorial plane or metaphase plate. So this is metaphase. Now take note that the word equatorial plane is used in KSSM syllabus. Metaphase plate is used in KBSM syllabus, but they're both correct. So use the right terms according to the ones used in your textbook. Okay, for KSSM is equatorial plane. The plane in the center of the cell is called the equatorial plane. Right, now you're asked to arrange them in the correct sequence. So once you've identified the phases, it's very easy. Prophase begins. So A, prophase is the first stage of mitosis, followed by D, metaphase, and then C, anaphase, and B, telophase. So the right sequence should be A, D, C, B. Okay, so all these cells look like some of the cells that you'll see under the microscope when you are looking at the real cells of animals or plants that are undergoing mitosis. In this case, it's an animal cell because you can see the cleavage furrow forming here. So 1 mark each for every phase and 1 mark for the correct sequence. So total is 5 marks. Let us go on to section B, which contains 6 objective questions. So each question is 1 mark. So the total number of marks is 6 marks. Now, you can read through the questions first, and you can also pause the video, and we will discuss the answers in about in a few minutes time. So pause the video, and you should take no more than 1.5 minutes to answer each question. Okay, for me, I will just wait a while and then I'm going to give the answer. Okay, so if you need more time, please pause the video or screenshot the question first and then refer to it. Okay, answers are going to be given right now. 3, 2, 1. Let's go. Now, the diploid chromosomal number of an organism is 16. What is the number of chromosomes in a somatic cell, a gamete, and a zygote of this organism? Okay, so diploid chromosomal number is 16. That means 2n equals to 16. 2n equals to 16. It means that for a diploid cell, which has 2 sets of chromosomes, the number of chromosomes is 16. Each set has 8 chromosomes. One set from the father, one set from the mother. So 8 times 2 equals to 16. What is the number of chromosomes in a somatic cell? Now, somatic cell is also a diploid cell. It has 2 sets of chromosomes, or the chromosomes, there are 2 copies of each type of chromosome. So somatic cell will also have 16 chromosomes. Now, gamete is a haploid cell, not a diploid cell. That means it has only 1 set of chromosomes. 1 set of chromosomes equals to 16 divided by 2 equals to 8 chromosomes. Okay. Now, a zygote is formed when a sperm fertilizes an ovum. That means 1 set of chromosomes from the sperm already combined with another set of chromosomes from the ovum. So a zygote is actually a diploid cell. It has 2 sets of chromosomes. Alright? So it should not have 8 chromosomes only. It should be the diploid number of chromosomes, 16. So the answer is C. 16, 8, 16. Because zygote is a diploid cell, and somatic cell is a diploid cell. Gamete is a haploid cell with only half the number of chromosomes in the diploid cell. Now for number 2, chromosomes uncoil and form fine chromatin threads. Spindle fibers disappear. Okay, the events above occur during mitosis, that is during which phase? So chromosomes uncoil and form fine chromatin threads. This occurs at the end of mitosis, and spindle fibers disappear. They are formed and then they disappear. So that's the end of mitosis. So it occurs during telophase. Answer is D. So first answer is C, second answer is D. Alright, so you can mark your answers too and see whether you got it right. Now let's continue with question 3. The diagram below shows a cell during a stage in mitosis. Which statement is false about the cell? So you can pause the video or screenshot this question and look at it. Pause the video and take about 1.5 minutes for each question at the most. And we'll come back to the question in a short while. As for me, I will just continue after 10 seconds. Alright, so let's look at this question. Now, which statement is false? So the cell has 4 chromosomes. 1, 2, 3, 4. There are 4 chromosomes, is correct. Each X-shaped structure is 1 chromosome. So this chromosome consists of 2 sister chromatids, but it's still considered 1 chromosome. So there are 1, 2, 3, 4 chromosomes. This statement is correct, so it's not the answer. There are 4 pairs of homologous chromosomes for this choice, B. Now, a pair of homologous chromosomes are chromosomes of the same size and they carry the same type of genes. There are 2 copies of each type of chromosome. Okay, so here we have 1 copy of the long chromosome. White color means that it may be from maybe from one parent, maybe the paternal chromosome from the father, and the black one, also of the same length or size, is the maternal chromosome from the mother. So this long black chromosome and long white chromosome forms a pair of homologous chromosomes, and the other pair is the short one, short white chromosome, short black chromosome. So there are 2 pairs of homologous chromosomes here. So this statement is wrong. Instead of 4 pairs, it should be 2 pairs of homologous chromosomes. So this is your answer. It's the false statement, B. Now for C, for the 2 daughter cells produce 2n equals to 4. So if we want to know how many chromosomes are found in the daughter cell, we must look at the parent cell. So in this cell, 2n equals to 4, okay, because this is a diploid cell, where there are 2 copies of the long chromosome and 2 copies of the short chromosome. So if it's a diploid cell, then what is the diploid number? So we just count the number of chromosomes, and there are 4, okay. So daughter cells that are genetically identical to the parent cell will also have 4 chromosomes. So this statement C is correct. Huh? Now, the chromosomes are arranged randomly at the equatorial plane. Yes, this is true, because this is in the stage called metaphase, where the chromosomes are aligned or arranged randomly at the equatorial plane or the metaphase plate here. So the answer is B, a false statement. Number 4, at which stages of the cell cycle do the following events occur? So, firstly, the centromere divides during which phase? Anaphase. Okay, so it can either be the first statement here, the first answer here, this can be correct, or C. But we go further down to this second event. DNA mass is doubled. Now the DNA mass is only doubled when the DNA replicates or the chromosomes duplicates. This occurs during the S phase of interphase. Okay, anaphase, interphase. So A is out, anaphase, prophase is wrong. So DNA mass is doubled during S phase of interphase. Thirdly, spindle fibers begin to form. This occurs during prophase, the first phase. So answer is C, anaphase, interphase, and prophase. How did you fare in this? Answer is 3B and 4C. 3B and 4C. Okay. Let us now look at question 5 and 6. So you can screenshot the question, or you can pause the video for a while and try to get the answers in 3 minutes, because each question takes about 1.5 minutes. The maximum amount of time is 1.5 minutes to try each question. So in total, you have 3 minutes. Then after that, we will continue by discussing the answer. So you can pause the video if you like. Okay, let's look at the statements. Which statement is true about mitosis and cytokinesis in cells? In the animal cell, centrioles form spindle fibers during mitosis. Correct. Animal cells have centrioles and they form spindle fibers during mitosis. Plant cell, centrioles do not form spindle fibers during mitosis. Plant cells don't have centrioles, so this statement is wrong. We are trying to find the correct or true statement. So highlight this if it comes out in the exam. Whether it's true or false, you have to highlight it. Make sure that you're not confused. Now we're looking for the correct statements. So this statement for plant cell is wrong. Therefore, the answer is not A. B, microfilaments contract. Yes, in cytokinesis, in animal cells, microfilaments will contract in the center of the cell. Plant cells, plasma membrane contracts. Wrong. Plasma membrane does not contract. It's just the formation of a new cell wall in the center of the cell. So B is wrong. C, plasma membrane splits. Not correct. It's the cell that splits into two as the cleavage furrow deepens. So if we say plasma membrane splits, tends to imply that the plasma membrane is torn or broken. This is not very correct. Plant cell wall is formed. Correct. But the first statement is wrong. So C is not the answer. Now D, cleavage furrow is formed. Correct. During cytokinesis in animal cells, microfilaments will contract, and when they contract, they form a cleavage furrow, which is a groove that deepens until the cell splits into two. Correct. Cell plate is formed. Yes, cell plate is formed from the vesicles that combine together in the center of the cell. And then from the cell plate will develop into a cell wall. Okay, so answer is D, the correct statement. Right? Number 6, the diagram on the right shows a cell during mitosis, which daughter cell is produced right at the end of the cell division. That means immediately after the cell division, after mitosis and cytokinesis. So referring to this parent cell, there are 1, 2, 3, 4 chromosomes in this parent cell. Then during mitosis, in anaphase, the sister chromatids of each chromosome will separate from one another and move to different poles and will be found in different daughter cells. So to determine what is in the daughter cell, just take one sister chromatid from every chromosome. So one long white sister chromatid, one long black sister chromatid from here, one short white sister chromatid, and one short black sister chromatid. And all these chromatids will then become a chromosome each at telophase. So this is the answer. Can we choose C as the answer? No. Immediately after the cell division, we don't have this C. We don't have chromosomes with two sister chromatids still attached because the sister chromatids have separated from one another and moved to different poles. Now, stage C only occurs when the cell in A goes on to the next cell cycle. It starts to go through G1, and then in S phase, it will duplicate. From this stage, it will duplicate to form this condition. All right, but not immediately after the cell division. You don't get this after cell division, immediately after cell division. You should draw chromosomes made up of single strands of DNA, okay, that have not duplicated. The DNA have not replicated, or the chromosome has not duplicated, okay, yet, until it goes into the next cell cycle. Let's continue with the structured questions. So once again, you can read through the questions here. You can screenshot the page and you can try the questions. Total number of marks is 19 marks. Okay, so for this page, maybe you can take around 10 to 12 minutes to try it. And then we will go on to the answers in the next slide. Now let us discuss the answers, okay, for the structured question. Number 1, the diagram below shows 3 cells of an organism X at different stages of mitosis. So you're asked to arrange the cells in the right sequence in mitosis. So first of all, identify the phases of the cell. P is telophase, as we can see that 2 new nuclei are formed, okay, and the sister chromatids have separated and moved to the opposite poles, so they are now single strands. They form the single strands of chromosomes. They become the daughter chromosomes. And then the cell is dividing into 2. That's cytokinesis, which occurs simultaneously with telophase sometimes. Okay, so this is telophase. And then prophase. This is prophase because you can see that the nuclear membrane is starting to disintegrate. Centrioles are moving to opposite poles and the spindle fibers are just forming. Okay, and also the chromosomes, the chromatin has shortened and thickened to form the chromosome structures. Now R is definitely anaphase because we see the sister chromatids have separated and are moving to opposite poles. So if you are asked to arrange the cells in the right sequence, that means you must start with prophase, then of course there's metaphase, but it's not shown here, then R anaphase and P telophase. QRP. Now, explain the events that occur at stage Q. 4 marks. So think of 4 events, 4 things that happen. At least 4 must be written. Chromatin shorten and thicken to form chromosome structures here. Nuclear membrane disintegrates. Nucleolus disappears. 2 pairs of centrioles move to opposite poles of the cell, and the spindle fibers start to form between the centrioles. So each sentence is 1 mark, and you can write more than 4, so that in case one of it is not correct, you will still have other answers that can help you to obtain the marks, the full marks. Okay, so there are 1, 2, 3, 4, 5. 5 events that happen are written here. C. State the behavior of the chromosomes at stage R and P. So at R anaphase, the centromeres divide into 2 sister chromatids, separate here, and when spindle fibers shorten and contract, they pull the sister chromatids to the opposite poles of the cell. Okay, this is all 1 mark, so we can either have the first part, centromeres divide into 2 and sister chromatids separate, or you can write when spindle fibers shorten and contract, sister chromatids separate and are pulled to opposite poles of the cell. Okay, either one. Now for P, telophase, we can either write chromatids arrive at the opposite poles, or chromosomes uncoil and form fine chromatin threads. I would prefer the second answer, okay, as the answer, right? So 1 mark each for R and P. So anyway, I think both answers should be able to be accepted for P. Okay, so let's go on to the next part. And do mark your answers. Try to give marks for your own answers. This is the last part of the structured questions, right? So again, read through the questions. You can screenshot and pause the video. And we'll come to the answers in a short while. So you have about 10 minutes to try this section. Let us look at the answer for question D. Label the parts of the cell as shown in R. So A is a pair of centrioles. B is a chromatid, not a chromosome yet. Okay, the sister chromatids separate and they become daughter chromosomes when they reach the poles. So it's better to label this structure as a chromatid, not a sister chromatid, because you must have 2 sister chromatids labeled to be able to label them as sister chromatids. If there's only one, we call it a chromatid. And this is the spindle fiber, right? So 3 marks, 1 mark for each label. Now E, based on cell R, draw cell S, which is in the phase before R. So the phase before R is metaphase, because R is the anaphase stage. So we are supposed to draw a cell S, in metaphase, but it must be based on cell R here. So you have to see which chromosomes are arranged at which chromosomes should be arranged at the metaphase plate, according to the arrangement in anaphase. You can see here that the first chromosome on the left is the long black chromosome. So draw the long black chromosome before the sister chromatids separate. So join them back together, like you are reversing the motion. So let them join back to each other. So this is a pair of long black sister chromatids joined at the centromere before anaphase. Now the second one is the short white chromosome. Okay, the third chromosome should be the short black one, and the fourth one is the long white one. Okay, just reverse. Imagine in your mind that this is replaying but in the reverse. The video is replaying in the reverse. So join them back together. Join back the sister chromatids together to each other. Okay? Then you can label some of the parts, because sometimes there are marks for labeling even though it's not stated. So you can label spindle fiber, chromatid, and centrioles. So let's say 1 mark is allocated for the labels. 2 labels equals to 1 mark. So if you have only 1 label, you don't get any marks. We don't have half marks for questions. It's either 1 or 0 mark. So 2 labels equals to 1 mark, and the other mark is for the drawing plus the correct position of the different chromosomes. Okay? Right, for question F and G, the answers are on the next slide. So we will refer to the next slide. Now, E is supposed to be a HOTS question here, okay, because you have to base your drawing of metaphase on the drawing of R, okay? So let's look at another HOTS question later on, okay. So these are the answers. What is the importance of mitosis in the production of large quantities of plantlets in agriculture? Now, if you analyze the question, it's talking about the importance of mitosis, production of large quantities of plantlets. So if we refer back to our lesson or notes, we know that the production of large quantities of plantlets occurred in the technique called tissue culture technique. So tissue culture technique is based on the process of mitosis, and it is used to clone, that means to produce large quantities of plantlets which are genetically alike to the parent plant, which has good characteristics. That they are chosen because they have good characteristics. Okay, so now the main importance of mitosis here is that it has an important characteristic, that is, daughter cells produced are genetically identical to parent cells. So if we carry out tissue culture involving mitosis, mitosis will ensure that the good traits of the parent plant is inherited by the offspring, that means by the plantlet or the clone formed. So a clone is an offspring that is genetically identical to the parent plant, 100% identical to the parent plant. And this can only happen if we allow the clone or the plantlet or the offspring to be produced through mitosis, not through sexual reproduction, but through mitosis. That means we take out some tissues from the parent plant, like the meristematic tissues, and then we put it in the culture medium outside the body of the organism, and we allow it to divide by mitosis. So all the new cells produced that form the plantlet will be genetically identical to the parent plant where the tissues come from. Okay, so this will ensure that the good traits of the parent plant is inherited by the offspring. Okay, that is what is that's the aim of producing the plantlets, is to make sure that they have good traits like high yield and high resistance to disease. Okay, so important points are the ones in color. First point, second point in green, third point here in purple, and fourth point is in red. Right, G, the diagram shows a cell that is undergoing mitosis. Here, spindle fibers T failed to form and did not join to W. So these spindle fibers here did not form, whereas the other spindle fibers did form. Draw the 2 daughter cells that are formed at the end of this cell division. So if T does not form, that means this pair of sister chromatids did not separate from each other and move to opposite poles of the cell. Okay, because T is used to pull the sister chromatids to different poles. So instead, this black chromosome will move to only one pole. The whole chromosome will move to one pole. The 2 sister chromatids will move to one pole. Whereas for the Y chromosome here, there are spindle fibers, so the sister chromatids will separate and they will move to opposite poles, okay, in the formation of the two new daughter cells. Thus, the first daughter cell will have one white chromosome when this chromosome is put to this pole. Alright, then the second daughter cell on the right hand side will have this whole chromosome, which is made up of two sister chromatids and also a white chromatid or chromosome, right? So this is your answer. So these two cells are not normal cells. They have undergone some mutation in that sense, and they are not genetically identical to the parent cell. How did you fare in the assessment given just now? Continue to try more questions on this topic, and you will become a real expert at this topic of mitosis. Now, in the next lesson, we are going to discuss a new subtopic, which is 6.3, meiosis, part 1. So meiosis is different from mitosis, right? So we're going to discuss what meiosis is, where does it occur, and why it is needed. So thanks for watching and stay tuned for the next video. Share, like, and subscribe. Goodbye for now.